Hello everyone, welcome to Raaonline. Today we will discuss our new chapter, that is biomolecules, right? As we know that biomolecule, what is that biomolecule? That is the term which is used for the molecules which are present in the living organisms, right? And they are essential to one or more biological processes, right? They are essential to one or more biological processes, like in biomolecules we'll study about the biomacromolecules, the biomicromolecules, right? Biomacromolecules, those molecules which is necessary necessary in large amount, right? Like the carbohydrate, the protein and the fats, right? So in that chapter we will study about their chemical structures, right? Their properties. Clear? All these things are studied under this chapter, that is biomolecule. Structure of the protein, primary structure, secondary structure, tertiary structure, all these. Clear? So come to the we will come to the summary of biomolecules in the form of questions, right? Come to the first question: which one of the following is not strictly a biomacromolecule, right? When we talk about the biomacromolecule, that means the molecules which are required in large amount, right? In large quantity. Clear? So when we talk about that, the protein, the carbohydrate and the fats comes under it, right? But when we talk about the the protein, lipids, polysaccharides and the nucleic acids, so lipid is not strictly a biomacromolecule. Clear? It's not, lipids are not strictly the biomacromolecule. So the answer is 2, because some of the lipids which helps in the formation of the steroidal hormones and like that, right, they comes under that the micromolecule, not exactly the biomacromolecules, right? The secondary metabolite that acts as a toxin is the aberrin, right? That aberrin is toxic for the body. Now the question is, what is that aberrin? That aberrin is a natural, it is a natural poison, right, which is found in seeds of a plant, right, called rosary pea or the Jequirity pea, right? Actually these peas are the red with black spot covering, right? These seeds are the red with a black spot covering, right, on one end. So aberrin is similar to the ricin, and it acts as a secondary metabolite, and it is a toxin, right? It acts as a toxin. Clear? So that's why the aberrin is the answer, right? Hello everyone, come to our new chapter, that is biotechnology, right? First chapter in biotechnology is principles and the processes, right? What is biotechnology? As its name suggests, bios, that is living, and technologies, right? So biotechnology is the technique of using living organisms or the enzymes, right, from the organisms to produce the products and the processes, right? To produce the products and conduct the processes which are useful for the humans, right? So that's why that branch is called the biotechnologies. We used that technology for making the curd, for making the bread, the wine, the batters of the idli and dosas, right? All the microbes mediated processes, right? These all are the microbes mediated processes, right? So it is used in restricted sense today. Why? To refer to such of these processes which use genetically modifying organisms, right? So we will study about the restriction enzymes, the plasmids, the vectors, right? The PBR 322 and all these processes, DNA fingerprint, DNA extraction, isolation, right? All these things comes under that chapter, right? We will study in the form of the questions. So come to the first question. Molecular seizures are, as we know that molecular seizures, that is the restriction enzymes, right? Restriction enzymes, the word molecular seizure and the chemical answer is A, right? It is also known as the chemical seizure or chemical knife, right? It is also known as the chemical seizure or the chemical knife, right? In restriction enzyme, DNA ligase, that means ligate, that means they attach something, right? So ligase enzymes, DNAs and RNAs and all. So the answer is the restriction enzymes, which are used as a molecular seizure to cut some part of the in of the in the organism's DNA, right? And retrieve it and implement it into the host cell, right? So when we talk about the restriction enzymes, actually restriction enzymes are the proteins, right? They are the proteins that recognizes a specific short nucleotide sequence, right? That recognize a specific short nucleotide sequence. And what will happen now after the recognizing? Cut the DNA, right? Cut the DNA from what at what place or from what particular site? That cut the DNA only at the specific site where the required gene is present, right? That's why it is called the these sites are called the restriction sites or the recognition sites or the target sequence, right? So these restriction enzymes are one of the class of the broader endonuclease group of enzymes, right? These are called the molecular seizures, these are called the chemical seizures, these are called the chemical knife, right? To cut the DNA, all the restriction enzyme makes two incisions, right? Two kinds of incisions, once through the sugar phosphate backbone and the another one from the DNA helix, DNA double helix, right? So that's why these restriction enzymes are also known as the molecular seizures or the restriction enzyme, chemical seizures and the chemical knife. Now come to the next question. Which enzyme joins the cut end of DNA? As its name suggests, as we know that restriction enzyme cut the DNA and the joining function is the function of the ligase, not lyase. That's why it's not answer. Ligase, ligate, that means attached, right? So DNA ligate cut joins the cut end of DNA, right? Restriction enzyme, the function of restriction enzyme to cut the DNA, right? And for one thing more, restriction enzymes are obtained only from the prokaryotes, right? They are obtained only from the prokaryotes, and that DNA ligase join the cut part of that DNA, cut end of the DNA, right? Now come to the next question. Below are given of the genetic engineering, arrange them in a proper order. All these are the lines of the NCERT, right? Identification of DNA with desirable gene. Okay, second is maintenance of introduced DNA into the host and transfer of DNA into the progeny. And third one is introduction of the identified DNA into the host, right? As we know that first we have to identify that particular DNA with the desirable gene which that type of character we want, right? So we have to identify that. After that, what will happen? We have to introduce that identified DNA into the host cell, right? And after that, we have to maintain that introduced DNA into the host cell and transfer of that DNA into the next generation, or we can say to the its progeny, right? So in that manner, the sequence is 1, then 3, then 2. So 1 3 2, 1 3 2 is answer is Bombay, right? The answer is Bombay. Clear? Come to the next. In 1963, the two enzymes responsible for the restricting the growth of that bacteriophage, right, in E. coli, which were isolated, what are the functions of these enzymes, right? So first one, that what is that bacteriophage? That is the disease causing, right? That bacteriophage is disease causing in the prokaryotes, right? What will happen here? Adding the methyl group to DNA, okay, that methyl group to DNA, that is called the methylase, right? It is also known as the methylase. Joining the cut ends of DNA. Cut ends of DNA? No, because it is the function of the DNA ligase, right? But we in that question, the two enzymes responsible for restricting the growth, right? Restricting the growth of, and that is the function of the restriction enzyme, right? This is the function of the restriction enzyme. So that's why that option is wrong. Adding formal group of DNA and cutting the DNA, no, that is wrong. Adding methyl group to DNA, that is methylase, and cutting the DNA, that is right, right? That is the function of the restriction enzyme. That's why we called, because they cut the DNA, that's why it is called the molecular scissor, na? Removing formal group and joining, no. So the answer is C, right? Come to the next question. First restriction endonuclease was, now what is that endonuclease? As we know that restriction enzymes belongs to a major group, right, of the nucleases, which is divided into two, that is endonuclease and the exonuclease, right? How we differentiated endonuclease and exonuclease? See that, right? That is a DNA template. When we cut the DNA from the terminal ends, right? When we excise from at the terminal ends, that is outer side, then it is called the exonuclease, right? And but when we excise in between, right? That is endon, within, between the mid portion, right? Then it is called the endonuclease. So when we talk about the first restriction endonuclease was, it was the HIND2, right? It was the HIND2. What is this, right? It was the it was found that actually that HIND2 always cut the DNA molecules at a particular point, right? By recognizing a specific sequence of the 6 base pairs, right? And this specific sequence is known as the recognition sequence of the HIND2. How we denote at what is the meaning of that HIND2? First letter, that first represents the genus, right? It represents the genus, and H for the Haemophilus, IN represents the species, right, which is known as the influenza. D, it shows the strain. 2, that is the Roman number in order to form, right? That is the Roman number in order to form. Clear? So this is called that is the way to represent a endonuclease, restriction endonuclease, right? Come to the next. Restriction endonuclease works on a specific nucleotide sequence, right? Restriction endonuclease actually works on a specific nucleotide sequence of the base pairs that read same on the 2 strands of the DNA when the orientation of reading is kept there, right? So these sequences may range from 4 to 8 nucleotides in length, or we can say, right, so that is called the palindromic sequence. That is called the palindromic sequence from any, that is from 5 to 3 or 3 to 5, right? When we read that, it read as same. In NCERT, example is given as Malayalam, right? When we read from that particular end, MALAYALAM, right? And when we read it in opposite direction, same as that, MALAYALAM, right? So this is called the palindromic sequence. This is called the palindromic sequence, right? For example, the GA-TTC, that is the EcoRI, right? TTC, right? So we know that C, G, T, A, T, A, right? A, C, A, T, A, T, G, C, right? So when we read it from that particular end, 5 to 3, that G, A, T, T, C, right? When here from 5 to 3, G, A, T, T, C, right? So this is called the palindromic sequence, right? This is called the palindromic sequence. See here, same as that, right? When we read it from that CAC, GATT, right? From that again, that is TTAC, TTAGC, right? So that is called the palindromic sequence. Which enzyme performs the above functions indicated by 4 small arrows, right? These are the 4 small arrows. That means restriction enzyme applied on that side and cut them from here, right? So what will happen? See, that is the that from that end it is, right, and from that end it. So what will happen now when we cut like that? These type of ends are called the sticky ends, right? These type of ends are called the sticky ends. When we cut like that, these are called the sticky ends, right? These sticky ends is also known as the cohesive ends. Clear? So what will happen? It shows the GAATTC, that is GA-TTC, that is the sequence of the EcoRI, right? That is Escherichia coli, rough staining and order is the first, right? So the answer is C. Clear? Come to the next. DNA fragments are separated by a technique, right, which is known as electrophoresis, chromatography, calorimetry, and by preparing hot extract of the solution. So that is called the gel electrophoresis, right? This is called the gel electrophoresis. DNA fragments are separated by the gel electrophoresis technique. So the question is now, what is that gel electrophoresis? Actually, DNA fragments is separated by that technique. We know that the DNA are the negatively charged molecules, right? That DNA are the negative charged molecule, and they can be separated by forcing them to move towards the anode, right, under an electric field through a medium, or we can say the matrix, right? So what will happen? The DNA fragments, nowadays the DNA fragments are separated according to their size, right, through the sieving effects which is provided by the agarose gel. Agarose gel is used in that process, that's why that process is known as the gel electrophoresis, right? Hence this similar, the fragment size, that further it moves with that, right? So what will happen here in gel electrophoresis? The separated DNA fragments, right, it can be visualized, only after staining. If we want to see these DNA strand without staining, so it is not possible, right? We will see just after the staining with a compound known as the ethidium bromide, right? After staining with, that is the question of pre-medical, after staining with the ethidium bromide, right? Ethidium bromide, which is followed by the exposure of UV rays, right? Which is followed by the exposure of the UV rays, because we cannot see the pure DNA fragments in the visible light and without staining, right? So what will happen now? We will see after the staining and come in the exposure with the UV rays, the bright orange colored bands of DNA, right? The bright orange color bands of DNA. Clear? The bright orange colored bands of the DNA in a ethidium bromide, right? Stained gel which is exposed to the UV light, right? So the separated bands of the DNA are cut from the agarose gel, right? What will happen after that? The separated band of DNA are cut out from agarose gel, right, and extracted from the gel piece, and that process is known as the elution, right? That process is known as the elution. This is also the question of pre-medical, right? That the separation of the DNA fragment from the gel piece after the staining, that process is called the elution, right? So after that, what will happen? We purified that DNA fragment in this way by using in constructing the recombinant DNA technology by joining them with the cloning vectors, right? So this is all about the gel electrophoresis, right? So that's why the DNA fragments are separated by the gel electrophoresis technique. Now during the DNA fingerprinting, the DNA is stained by chloramphenicol, phenolphthalein, ethidium bromide, all that used by the ethidium bromide, right? We already studied about that. The DNA is viewed under UV rays, IR, Webgyor, radio waves. So the UV, just we study about that in gel electrophoresis, that we'll see in ultraviolet radiations, right? That DNA is viewed in the UV rays. Now come to the next. What is elution, right? Elution, that is coating of DNA with microparticle of gold. No, that is gene tungsten and all these things, gene gun, that is a different thing, right? Participation of DNA using a suitable agent. No. Laminating DNA with suitable light. No. Cut out and extract the bands of DNA from gel. Exactly, elution is the cutout and the extract bands of the DNA from gel, right? Now come to the next. This sequence from where DNA replication start is known as the origin of replication, right? So the answer is the Bombay, right? That is called the origin of replication. Clear? So now what are these? These are this is called the tetracycline resistance, right? That is the antibiotic resistance, right? Tetracycline is an antibiotic, so that is the tetracycline resistance, which shows the antibiotic resistance. What is that rope, right? That rope is the codes for the proteins, right? These are called the codes for the proteins which are involved in the replication of the plasmid, right? Which are involved in the replication of the plasmid, which is called the rope, right? So ORI, that is the origin of replication. ORI is the origin of replication. Clear? Now come to the next question. Normal E. coli or the non-recombinant E. coli contains the resistance against ampicillin, tetracycline, both or none, right? When we talk about the normal E. coli, then it shows the it does not show the resistance against anything, right? So the answer is the none, right? A line is marked in NCERT that normal E. coli cells do not carry resistance against any of these antibiotics, right? So that means it doesn't show the resistance against the ampicillin and as well as the tetracycline, right? So next, come to the next, that is procedure through which the piece of DNA is introduced into the host bacterium, right? It is called transformation, biolistic, both, none. It is called the transformation, right? That process is known as the transformation. Clear? That process is known as the transformation now, but what is that biolistic? That biolistic is also known as the gene gun, right? That biolistic is also known as the gene gun, right? What is that biolistic or gene gun? It is the method for the delivery of nucleic acid by cells, right? Nucleic acid to cells, actually. Nucleic acid to cells by high speed particle bombardment. High speed particle bombardments, right? That technique is used the nucleic acid coated proteins, right, which is propelled by a pressurized gun, clear, which is called the gene gun, clear. So this is called the biolistic method or the gene gun method to transfer the cells or the organelles, right? It can also be used to deliver the vaccine as well, clear. So these are called the biolistic or the gene gun, clear. Come to the next. If rDNA, what is that rDNA? That is recombinant DNA, right? That rDNA is the rDNA, that is recombinant DNA, clear. That rDNA is introduced in a gene coding for tetracycline, right? It is introduced in gene coding for the tetracycline. Then the host will become resistant for tetracycline? No. The host will lose the resistance for tetracycline. Exactly, when we introduce the gene coding for the tetracycline, then what will happen? It doesn't resist the tetracycline because we introduce into the DNA, right? Recombination of DNA. The host will start producing tetracycline. No. So the answer is the Bombay, right? The host will lose the resistance for the tetracycline if the recombinant DNA is introduced in a gene coding for the tetracycline, right? Now come to the next. Insertional inactivation is results in inactivation of cancer cell, oocyte, and enzyme, all, right? So it is used for, this results in inactivation of an enzyme, actually insertional inactivation, right? That insertional inactivation is what actually, right? So when we talk about that, that is the method, right? Insertional inactivation it is a technique, or we can say the method, right? It is a technique which is used in recombinant DNA technology, right? Recombinant DNA technology. What will happen in that case? In this procedure, a bacteria, right? In this procedure, a prokaryotic cell or a bacteria carrying the recombinant plasmid, right? Recombinant plasmid, or we can say a fragment of the foreign DNA, right? Or we can say a fragment of the foreign DNA which is made to insert into a restriction site inside a gene into a restriction site inside the gene, right? Why? To resist the antibiotics, right? To resist the antibiotics. So what will happen now? It causing the gene to turn non-functional or in an inactivated state, right? So that is called the insertional activation, right? So it results in the inactivation of an enzyme. Clear?